Climacteric and non-climacteric fruits differ significantly in their ripening processes, particularly in the roles of ethylene biosynthesis and signaling pathways. Climacteric fruits, such as tomato, banana, apple, and mango, are characterized by a sharp rise in respiration and ethylene production during ripening. In contrast, non-climacteric fruits, including grape, citrus, strawberry, and pineapple, ripen without a notable increase in either respiration or ethylene levels. This fundamental distinction shapes the molecular and physiological mechanisms underlying ripening in these two fruit categories. In climacteric fruits, ethylene plays a central role as a primary regulator of the ripening process. The biosynthesis of ethylene is significantly upregulated as the fruit transitions from unripe to ripe. Key enzymes such as 1-aminocyclopropane, 1-carboxylic acid, ACC, synthase ACS, and ACC oxidase, ACO, are activated, leading to the conversion of S-adenosylmethionine, SAM, into ACC, which is then converted into ethylene. This surge in ethylene triggers a cascade of signaling events essential for ripening. Ethylene binds to specific receptors such as ETR1 and activates downstream signaling components, including EIN2 and EIN3, which in turn regulate the expression of ethylene-responsive transcription factors, ERFs. These ERFs control the transcription of ripening-associated genes involved in processes such as cell wall softening, pigment synthesis, and aroma development. This ethylene-driven mechanism ensures that climacteric fruits ripen uniformly and are responsive to external ethylene applications, which can accelerate ripening. Non-climacteric fruits, on the other hand, exhibit a fundamentally different ripening mechanism. In these fruits, ethylene production remains minimal, and enzymes like ACS and ACO show low activity during ripening. Consequently, the ethylene signaling pathway is either inactive or plays a minor role. Instead, ripening in non-climacteric fruits is regulated by other hormonal and molecular factors, such as abscisic acid, ABA, auxins, and sugars. These signals orchestrate the biochemical and physiological changes associated with ripening, including color development, sugar accumulation, and texture modification. Unlike climacteric fruits, non-climacteric fruits do not respond significantly to exogenous ethylene treatments, highlighting their ethylene-independent ripening pathway. This characteristic also contributes to their longer shelf life and greater storage stability compared to climacteric fruits. The distinction in ethylene biosynthesis and signaling pathways has significant implications for the regulation of ripening genes in these two types of fruits. In climacteric fruits, the expression of ripening-related genes is heavily dependent on ethylene. These genes regulate key processes such as fruit softening through the degradation of pectin, color changes due to the breakdown of chlorophyll and synthesis of pigments like carotenoids, and the development of characteristic flavors and aromas. In non-climacteric fruits, similar processes occur but are driven by non-ethylene pathways. For instance, ABA and sugar signaling play a critical role in regulating the ripening-associated genes in grapes and strawberries. The practical implications of these differences are also notable. Climacteric fruits, being ethylene-dependent, can be harvested before full ripening and artificially ripened using ethylene gas, allowing for greater control over their ripening time and reducing post-harvest losses. However, their reliance on ethylene often leads to rapid ripening and shorter shelf life, posing challenges for storage and transportation. In contrast, non-climacteric fruits ripen more slowly and maintain their quality over a longer period, making them more suitable for prolonged storage and distant transportation. In summary, climacteric and non-climacteric fruits represent two distinct ripening paradigms shaped by the role of ethylene. Climacteric fruits depend heavily on ethylene biosynthesis and signaling, with this hormone serving as the central regulator of their ripening processes. Non-climacteric fruits, in contrast, follow an ethylene-independent ripening pathway controlled by alternative hormonal signals. These differences underscore the complexity of fruit ripening and provide valuable insights into their post-harvest handling, storage, and commercial applications.